Hello, my name is Jake DeMillemeester. I'm a student at Wayne State University studying biomedical engineering with a concentration in biomaterials. So today's topic has a lot to do with biomaterials. It's how research is done on new drugs on human body tissues. So today we'll talk about how drugs are researched now and how they affect tissues, what an organ on a chip is, and the benefits that organ on a chips have, such as accurate results, connectivity, and personalization. So what's currently being done in labs is drugs are tested on lab-grown cells in petri dishes or vials, and after that initial test is successful, they're tested on lab animals and eventually on human trials, and then eventually released to the market. This process takes a lot of time and is very expensive, and not many drugs are actually output using this method. Only 1 in 10 actually get to human trials. So an organ on a chip is a new method to research how drugs affect actual human cells. It's the size of a credit card with plastic molds and chambers inside connected through channels. And what happens is human tissues are placed inside each of the chambers, and a fluid filled with nutrients and oxygen is circulated throughout the channels and into the chambers, along with the drug that's being tested. So this method has much more accurate results than on the cells done in petri dishes, because these cells the cells grow in a way more similar to how they are in the human body because of the circulation of fluid. When the same toxin was exposed to cells from petri dishes and from the organ on a chip, the organ on a chip released a biomarker indicating injury, while the petri dish cells did not release anything. Another benefit is that the organ on a chips can be connected together with different types of cells, which would simulate how a drug can affect different parts of the body, such as the team at the University of Washington connected liver cells to kidney cells to analyze how they would be affected by a single drug. And these chips can be personalized to a certain patient. So a patient's own cells can be placed inside the chip, and then a drug can be exposed to those cells to see how a patient's own body would react to those without having to have the patient take the drug and affecting the whole body. This saves the patient a lot of time and money and reduces risk to the patient by not having them take a pill that might not be as effective as another one. So in total, organs on chips have the potential to decrease research time, save money to the patient and pharmaceutical companies. They can test tissue that more closely resembles the actual human body tissue. They can be connected to see how a drug affects multiple organs and organ systems, and they save the patient time, money, and decrease risk to that patient. Thank you.